Rebecca Craighill Lancefield was born in Fort Wadsworth, New York in 1895. She attended Wellesley College in Massachusetts and eventually graduated with her bachelor's in zoology, although she was originally set on being a liberal arts major. After accepting a scholarship, Lancefield attended Columbia University for her teaching, but petitioned the university to allow her to pursue her education in bacteriology. After graduating, she became a laboratory technician at the Rockefeller Institute for Medical Research. Working alongside Oswald Avery, their research began upon the study of streptococcal bacteria. After this time, she later worked in a laboratory at Columbia University, but was rejected for their PhD program for being a woman. She eventually accepted a second job, working at Rockefeller University again. After some time, Columbia University eventually granted Lancefield her PhD, and she continued working at Rockefeller University for the remainder of her life. Her main work focused around the classification of different streptococci. Lancefield eventually passed away in 1981 at the age of 86 after complications of a hip fracture she received in 1980.
As a pioneer in microbiology, Rebecca helped classify and put forth research on the streptococcal bacteria that infects humans. During her career, Lancefield developed the Lancefield grouping system to classify bacteria. Lancefield later identified that the streptococcal bacteria was the leading cause of a number of diseases such as scarlet fever, erysipelas, or strep throat. She identified different surface proteins on groups of streptococci to learn more about the antigens present on them. Later, Lancefield was a member of the epidemiological board of the armed forces during World War II and focused on streptococcal and staphylococcal diseases during this time. Rebecca was also academically accomplished as she became a professor at Rockefeller University and Columbia University. Rebecca became interested in the study of streptococcal bacteria during World War I, as many soldiers and civilians became plagued with diseases such as scarlet fever and secondary streptococcal infections. Millions of children suffered debilitating heart diseases from rheumatic and scarlet fever, and the precise etiologies of these conditions were unknown. Rebecca wanted to find out what was causing issues among the soldiers and civilians and succeeded in developing a classifying system to help identify the Streptococcus bacteria. This was essential in order for the development of a vaccine that would lower the chances of contracting these diseases.
The M antigen antibody reaction is an example of the experiments she ran for classification of streptococci. During this experiment, Lancefield mixed the M and C antigens with a mixed growth and broth. After being centrifuged overnight, the mixture is heated with boiling water with the occasional shaking and cooled and then centrifuged again. The solution was colored yellow, and NaOH and HCl was added to the mixture drop by drop, and the mixture was then centrifuged again. The M antigen is added, and if a positive reaction occurs, the streptococci is classified as type M. If the reaction is negative, the mixture can be tested against other serums. However, this is not all Lancefield experimented with. She also discovered 2 different antigens of streptococci, one of which was related to the 1918 outbreak, a streptococcal infection that broke out during World War I, and the second is a feature related to all forms of streptococci. As the leading female researcher of her time, Rebecca served as a pioneer and role model for other females entering into biological research. Among her remarkable accomplishments, Lancefield was the first female elected to be president of the American Association of Immunologists, as well as president of the Society of American Bacteriologists. Lancefield's work helped lay down foundations for the epidemiology, biology, immunology, and pathogenesis of streptococcal infections. Lancefield's work has helped people classify different groups of streptococci, such as Group A and Group B streptococci, which affect the different groups of humans and bovines, respectively. Her classification system today has groups A through M and she is considered the streptococcal pioneer.